Thank you for the invitation. So I want to start by suggesting a hypothetical scenario. What if insulin resistance can be rapidly reversed in humans? I mean reversed, not a modest improvement. This is an important question because insulin resistance remains to be one of the biggest unsolved problems in metabolic diseases. And for decades, despite decades of research, no therapy can effectively and substantially reduce insulin resistance. In many ways, it has become the holy grail in metabolic disease therapy. That is why what we observed in our phase 1 study was so unexpected. In our phase 1 study, we, with only 14 days treatment with LP01E or compound, we observed a rapid and substantial reduction in multiple insulin resistance-related indices in type 2 DM. And to be honest, this is not what we expected to see initially. To address insulin resistance, Alphala developed LP01E, which is a small molecule compound already passed phase 1 trial and entered phase 2 trial in the United States with FDA clearance. So as you might know, the type 2 DM is driven by relative insulin deficiency and insulin resistance. Insulin resistance further results in compensatory hyperinsulinemia and progressive beta cell stress. Hyperinsulinemia further worsens insulin resistance, generating a vicious cycle, drive disease progression. Currently, most treatment for type 2 DM focus on relative insulin deficiency and downstream hyperglycemia. So if you think of type 2 DM as a fire, the insulin resistance is the fire, the root cause of disease, and the hyperglycemia is the smoke, the symptom we see clinically. If you only treat hyperglycemia, if you only remove the smoke, the fire continues to burn. If you only reduce blood glucose, the disease continues to progress, and ultimately you are not able to change the natural course of type 2 DM. Instead, our approach is focused on the root cause of the disease, insulin resistance itself. So LP01E is a small molecule compound, and it is a novel small compound targeting the upstream biology of insulin resistance. And in our phase 1 study, we observed something unexpected. So based on published literature, around 24% of HOMA2IR. HOMA2IR is a marker to measure insulin resistance. This is published data. The best data show that 24% of HOMA2IR can be reduced with after 40 weeks tirzepatide, maximum dose tirzepatide treatment. Tirzepatide is a popular weight-losing drug. Yeah, and for 40 weeks. But in our phase 1 study, we observed a 40 to 50% reduction in HOMA2IR within just 2 weeks. So the magnitude and the speed of this drug effect appears difficult to reconcile with what has been reported for the existing therapy. So this result immediately brings me the next question. Is it real or it is just an artifact? Okay. So the gold standard to measure insulin resistance is euglycemia clamp. However, it is too complex to be used in a phase 1 trial. So therefore, we use multiple validated insulin resistance related indices, no matter it is vaccine-based or it is OGTT-based, including HOMA-IR, HOMA-S quick I, and HOMA2IR, and the GUTS index. Matsuda index and OGIS 120. So the data were striking. We found a consistent improvement in those multiple insulin-resistant related indices. And in addition, while blood sugar, because LPS1 also potently reduced blood sugar, we found while blood sugar show a great reduction, the fasting and the post-OGTT serum insulin remain unchanged. So you know that the body needs insulin to reduce blood glucose. So why don't the insulin level remain the same, but the blood sugar level greatly reduced? Furthermore, we found the drug effect means that the level of HOMA2IR improvement is highly correlated to the level of underlying HOMA2IR. So this is another data showing, this is the data of OGTT-based Matsuda index. It is insulin sensitivity index. The higher the index is, the better the insulin sensitivity is. This data showing that our drug can efficiently increase Matsuda index up to 70% only after 2 weeks treatment. So from safety perspective, LPS1 demonstrate an excellent safety profile. In our phase 1 trial, all treatment emergent adverse events were mild in severity and with no significant difference in their incidence rate between LPS1E and the placebo. So from efficacy perspective, LPS1E demonstrate in our phase 1 trial, because in our phase 1 we not only recruit healthy volunteers, we also recruit type 2 DM patients. We demonstrate that we have potency to reduce insulin resistance. And we also demonstrate that we have strong potency to reduce blood glucose. We can reduce HbA1c within just 2 weeks treatment. Everybody knows that A1C represent is an indicator to evaluate average blood sugar within 3 months. But we can reduce A1C only after 2 weeks treatment. And we also have a potential to improve beta cell function. We strongly improve HOMA2 beta. So with the potential to improve insulin resistance, it offers a possibility to move the treatment upstream to have an early intervention to treat pre-diabetes, which is a new therapeutic category, new and underaddressed therapeutic category with multi-billion-dollar potential. Okay. So because insulin resistance is a driver of multiple diseases, we believe this is a major therapeutic white space. And we believe in the future that treatment for type 2 DM require multiple therapeutic pillars. GLP-1 agonists treat weight, SGLT2 inhibitor treat organs. At Alphala, we aim to treat disease itself by targeting insulin resistance at its root. The data has been presented in the ADA meeting this year, and we just host a scientific discussion with Dr. Rolf DiFranzo and Zen Fleming in New Orleans. Thank you.
Thank you very much. Questions? If we hurry, we can get 2 questions in. Any questions?
Just curious, you said it's small molecule. How are you presenting it, tablet, capsule, knowing that the GLP-1s are typically injectable and people don't love to do that to themselves every day? So
yes.
Just curious what your presentation is.
Just pills.
Just pills?
Yeah.
Capsules, tablets?
Once or twice daily. Yeah, capsules.
Great.
Yes.
Any questions online?
Yeah, quick question. Where are you in the approval? Oh, okay, phase 1. That's where you are.
Yeah, we already passed phase 1 and got FDA approval to enter phase 2 trial in the United States.
Okay, thank you.
Thank you.
Last question.
So you didn't talk too much about how you can protect the drug after it releases, and if you do get approved, what's your IP moat look like? How do you protect the drug from being copied later on?
Oh, it's very difficult program. I'm happy to talk to you in details because we already out of time. Do we have time for?
Wrap it up. Whatever you're talking about, wrap it up.
Yeah, this is a very difficult question, and previously no drug. This is the first drug to improve insulin resistance, to my knowledge. First, the only, fast and substantial. Yeah, and it's very difficult to because it is unknown mechanism,
Okay.
and it's very tricky. Happy to talk to you about this more.
All right. Thank you to
Thank you.
Alphala.